Hello, everyone. This is Mr. Friedhoff. I'm going to go over chapter 1 in your AP biology book and probably every chapter there after this. I know with ending last year a little early, we didn't fully go through all your chemistry or whatever science class you had last year. So I'm just going to give you chapter reviews and go through the PowerPoint myself on here. For the first two chapters, I'll go through the whole PowerPoint, and then every chapter after, I'll probably do the sections. This first chapter is actually pretty easy. It's about basic science principles. Now, I say that's the most easiest, but it's probably one of the most important as well. We're going to spend a little time in chapter 1 just during the first full week of school. One of the big tips I'm going to give you as we go through this, start trying to break down words, especially with the first we're going to go over biology. It's really easy just to kind of go, oh, I know biology, and just move through it. Try to understand the prefixes and suffixes. When you get to my class, you're going to see one of my walls is all vocab words, and one of my walls also has prefixes and suffixes on them. Try to break down words using those prefixes and suffixes. I will also give you a big sheet with it at the beginning of the year. This helps during any test and just going forward in life. It really does help. All right, let's get started in chapter 1, A View of Life. Now, the first vocab word we're going to go over is biology. I know you've heard of it before, but again, bio means life. Ology means the study of. Once you put them together, it means the scientific study of life. Now, I know it's pretty easy just to memorize biology as the study of life, but once you get in the habit of breaking down words and their prefixes and suffixes, this becomes a lot easier. So start doing that automatically. What does each part of the word mean? The cells are the most basic unit of life. We're going to start in this class with stuff that's very, very small. So after we get through the scientific principles, we're going to start talking about atoms, about protons, electrons, neutrons, about chemicals, about compounds, about chemical bonding. And then we're going to move up to macromolecules, which are a little bit bigger. We're going to talk about cells then and organelles and how DNA within the cell regulates how an organism or what an organism looks like. So we're going to get bigger and bigger, and then we're going to talk about how species interact with each other and how species might evolve around each other or by each other, just in a mix together. So we're going to start out very small, and we're going to get to be very large in this class. So we're going to go, again, at the atomic level, and then we're going to look at the ecosystems in the world as a whole. And that's what it says down at the bottom. Biological organization shows how properties change as you observe different levels. So as we move up the different levels and as we get larger and larger and looking at the scope of biology larger or more broader, I should say, we're going to see these properties change. Now, the two main forms of cells. I'm going to say this time and time again. You're going to hear this 1,000 times, so just learn it now. Eukaryotic cells. There's two different types of cells. Eukaryotic is the first, prokaryotes the second. So eukaryotic cells. You are a eukaryotic cell. Think eukaryotic. Eu is also yes, it has a nucleus. Pro is no. Prokaryotic cells do not have a nucleus. So think U is yes, pro is no. U is yes, pro is no. Now, prokaryotic cells, they're also very small. If you look at our PowerPoint here, look how small this prokaryotic cell is. Very, very small compared to the eukaryotic cell. Each of these cells, or I should say all cells, have two main properties. One of those main properties is that they're enclosed by a membrane. So even though the prokaryotic cell, here we go. Even though the prokaryotic cell is very small compared to the eukaryotic cell, it still has a membrane. It looks different than the eukaryotic membrane, but it still has a membrane. The eukaryotic membrane also looks a little different than, again, the prokaryotic cell, but it's a lot larger. The last part here, all cells have some type of genetic material, either DNA, which most cells have, or RNA. So again, all cells have some DNA genetic material. Our next slide here, we're going to talk about unicellular versus multicellular. Uni meaning one, and then cellular is just how many cells it's composed of. You can see some examples here of unicellular organisms. Most prokaryotic cells are unicellular. I should say all prokaryotic cells are unicellular. There are some eukaryotic cells that are unicellular, but they're not there's very, very few of them. Most eukaryotic cells are multicellular. Our golden retriever here. But again, you want to start breaking down these words. I know unicellular and multicellular are pretty easy to break down, but once you get in the habit of breaking down these words, it's going to become a lot easier later on in the class. We're going to talk about that organization of life from two slides ago. Down here again, we're going to look at the atoms first, and we're going to see how they're going to make up like compounds, molecules. We're going to see how macromolecules start making up different organelles in the cells. We're not going to spend too much time on the tissues and organs and the organ systems. We're going to look at the organism. This block of biology is usually kind of focused more on the anatomical side of things. So if you're taking an anatomy class, this is where we're going to spend a lot of your time. We're going to kind of skip and go right to the organism and talk about how genes influence organisms. Then at the end of class, we're going to talk about how populations, ecosystems, and communities all kind of work together to create our biosphere. And look at just how evolution kind of dictates how life on Earth has evolved throughout billions of years. And that's like my favorite part of the class. In our next slide, we're going to talk about the three domains of life. These three domains are bacteria, archaea, and eukaryotes. Now, bacteria and archaea are single cell, which means they're unicellular, and they're prokaryotic. Remember, pro is no, u is yes. Oh, my lights just turned off. So what we're going to see is, again, bacteria is really common bacteria, just bacteria. Archaea is a very old type of kind of bacteria. You would think, like visually, when you're looking at it, it's very, very similar, and if not identical to bacteria. But when scientists were looking at the outside cell wall and membrane, they realized that it had enough difference and it had a different genome with it that we could classify it as a new domain of life. Now, plants, animals, fungi, and protists all belong to our last domain of life, which is called eukarya. Those are the eukaryotes. You can see examples here: the animalia, the fungi, the plants, and the protists. Protists are those single-cell eukaryotic organisms, and they're pretty interesting by themselves. But again, three domains of life: bacteria, archaea, and eukaryotes. Slide, our next piece of content are the characteristics of life. Now, I'm going to talk about something that I'm going to mention time and time again in this class. It's very hard to paint broad strokes. It means like one rule encompasses all of biology. Just because you'll write all cells have this or all organisms do this, and then you always find that one stinking organism that just doesn't follow that rule. Does it mean that rule doesn't apply at all? Not necessarily. It just means we have to have a conversation about the effectiveness of that rule. So it's very hard to kind of especially in biology, just because life is just so dynamic, to paint broad strokes of content. So why I'm telling you about this is here are 6 characteristics of life. I remember, I think 5 back when I was in high school. I've seen some textbooks have up to 7 or 8 characteristics of life, and they're all very similar, but different. Biologists will say, well, I don't agree with this, and they'll have arguments about the characteristics of life themselves. Now, does that mean that this is wrong? No, absolutely not. It's just all science will agree, yeah, there are characteristics of life. Which ones should we include in our list are just a little bit debated. So you can see here: acquire materials and energy, maintain homeostasis, respond to stimuli, have adaptations and evolve, reproduce and develop, and has a genetic code. Looking at those more closely: acquire materials and energy. That's just eating and breathing and things like that. Now, not all organisms will breathe, but I'm just giving you an example there. Maintaining homeostasis, that's just your internal mechanisms keeping you at certain levels like pH, temperature, water levels. Responding to stimuli, that's just the response to outside factors, like when it's really hot out, you start to sweat. Have adaptations and evolve. So all organisms evolve over time, and that's just another characteristics of life. We're going to talk about evolution a lot here. I think the next slide we're going to talk about evolution, but we're going to start talking about evolution today, and we won't stop talking about evolution until probably the end of the year. Reproduce and develop. So all organisms, kind of like, yeah, well, duh, all organisms have to reproduce and develop. Whether you're a prokaryotic unicellular organism, you're going to go through asexual reproduction, or you're a human, you go through a sexual reproduction. And all organisms have to have some sort of genetic code to pass on and to reproduce. Now, like I said from the last slide, biology is centered around evolution. I absolutely am just a geek when it comes to evolution. I took a class this past summer about dinosaur evolution. I absolutely love talking about evolution. It's just what I truly love about biology. But the first word or the first definition we're going to get is change over time. So I want to start there and start talking about evolution in each one of these chapters a little more, and we're going to see how the content really relates to evolution. One of the best quotes I have, it's actually on my teacher page, is by, I'm not going to try to pronounce this guy's name. I think he's Russian. But he was I think, a biochemist. And he said, nothing in biology makes sense except in the light of evolution. I remember hearing that in my high school biology teacher's class. And I remember rereading it in even college. And I really didn't understand how important that line is and how true that line is until I started teaching it and started really diving deeper into the material and understanding how true this is. You might not connect with it just yet. In 20 years or in 10 years or in 5 years when you guys are in college, you might be like, oh, yeah, I remember Mr. Friedhoff saying that line. I kind of get it now. So I'm going to start to show you how evolution is incorporated in every single chapter in this book. Our last section of chapter 1 that we're going to talk about is called the scientific method. Now, a lot of you have probably already seen this in some capacity back in even elementary school, but most definitely in middle school and in your college prep and biology classes and your chemistry classes and physics. You've seen this before. You know the whole process. What I want to pause here and do is make sure you understand how important this is. There's a lot of scientific research out there that shows if a student doesn't truly understand a scientific method and science, terminology and things like that, they're not going to truly understand, or they're going to have a tough time understanding things going forward. So to understand this first chapter, I'm telling you, it's monumental. It's very crucial for your understanding of the following content throughout the year. So don't just breeze through this part of the chapter and say, oh, I've done this before. I know what a hypothesis is. I know what a theory is. Make sure you take some time and truly understand this stuff, because it's crucial for your understanding of things going forward. Our first vocab word that we're going to talk about today is hypothesis. Now, a lot of you can probably already regurgitate and say, educated guess, and that's great. However, there's two main parts that I want to talk about that are really, they're really important when we talk about hypotheses in AP biology going forward. First is that it has to be testable and it has to be falsifiable. Testable means it has to be able to make sense and it has to have some type of experiment that can test it. And it also needs to be falsifiable. It needs to be able to be disproven. Now, I don't like using the P word. I don't like it saying proven in my classroom. It's kind of like a swear word because you don't prove things in science. You can say this is supported by all of the evidence in science, or this hypothesis is supported by this, this, and this. We don't say we prove things in science. That's a no-go in science, or at least in my classroom. You might see that in literature, in like the media, and they'll say, oh, scientists prove this. Again, it's different from what actually scientists say, but we don't like to say proven in my classroom. It's kind of like a swear word. And it also must be consistent with what's already known about the universe. So a hypothesis should be testable, it must be falsifiable, and it must be consistent with what we already know. The bell will ring at the end of the period. That's a great example of a hypothesis. We can sit here, we can wait for it, we can listen for it, we can get microphones that can test the decibel readings. We can test that hypothesis and we can falsify it. We can say, oh no, it didn't ring because of the bell system broke. A fact is different from a hypothesis because a fact is an observation that can be repeatedly confirmed. Now, don't get hung up on that one word in there, observation. Just because we can't see how hot the sun is, we can obviously look up at the sun, don't do it, but we can obviously look up at the sun and say, yeah, it's probably pretty hot. But we can't say, well, it's 10,000 degrees Celsius or 1,000,000 degrees Celsius. We have to use instruments and we have to use scientific ways of measuring the sun to see it's how many degrees hot. That doesn't mean it's not an observation. What that means is the observation comes from taking data and taking measurements from the sun. So an observation is just something that's repeatedly confirmed time and time again. The bell rang last period. Yeah, we can check to see if the bell rang last period. We can look at the video monitors. We can talk to people. We can take instruments and we can actually record the bell ringing. We can take multiple sources of information and multiple sources of observations and repeatedly confirm that the bell rang last period. One of the most important parts of this chapter is going to be this slide right here, where we're going to talk about the null versus alternative hypothesis. Now, if you talk to any of my AP biology students from previous years, they're going to tell you, you probably see these every other lab, if not every lab throughout the year. So this is really important. What I'm trying to get across is this is really important stuff going forward. And especially if you want to do a career in science, you're going to see this all the time in your research and in your college classes. So a null hypothesis, it's abbreviated HO, it states there's no significant difference between the observed and predicted values. What this means is when you set up an experiment, you're going to predict certain values. What the null hypothesis states is that there is no significant difference between those predicted values and the observed values, or what you actually record from your experiment. So you have your predicted values and you have your observed values. You have to compare them. And we're going to talk a lot about that this year, comparing the predicted versus observed values. If there is no significant difference between the observed and predicted values, that means that you fail to reject your null hypothesis. If there is significant difference between the observed and the predicted values, that means you reject your null hypothesis. I don't say accept, it's either fail to reject or you reject your null hypothesis. Your alternative hypothesis states that it's just your regular hypothesis. It's the hypothesis the researcher is trying to test and say, hey, this is what I predict is going to go on. But it's just like your regular hypothesis, and that's denoted with the H1. Now, to do this testing, you have to have controlled experiments, which means the variable is going to be unchanged, and you have to have a baseline that lets you see if the treatment is actually having an effect. And we're going to talk about that in our next slide. With control groups, there's actually two different types of control groups. You have a negative and a positive control group. Again, these are pretty significant, but they're not really like you have to like regurgitate the information and or define them word for word. But let's talk about them a little bit more here. A negative control group is an experiment where no response to the variable occurs. So what you're doing is you're checking for false positives. You're checking for if the experiment that you produced or the experiment that you're running is reliable. You're looking at, every variable, whether it, each variable is being tested and is the one affecting the whole system. Positive control groups are experiments that receives a treatment with a known result. This confirms that the system is working correctly. And by system, I'm talking about the experiment itself. All of the devices, all of the machines, all of our data taking, data recording instruments, if they're working correctly. We're going to talk about an experiment next that you're going to find online. If you look on YouTube and you search for positive versus control experiments, there's a nice 4-minute video that explains these. But I'm going to go over them pretty quickly here. So if we're going to talk about how coffee affects blood pressure, we're obviously going to check we're going to have several volunteers and they're going to drink coffee and see how it affects blood pressure. But we're also going to have to check for other variables within this experiment, meaning it's not just the coffee the person's intaking. They're also intaking water. They're also intaking maybe it's just the coffee itself. Maybe it's not even drinking the coffee. Maybe it's just, it's something else. So what we have to do is we have to have a bunch of negative controls. So you have to give people coffee. You have to give people decaf coffee. You have to give people water, and you have to let people not drink as well. So maybe it's the drinking of something that's lowering blood pressure. We don't know. We have to test each one of these individually. These are called negative control groups. Positive control groups, remember, we already know what's going to go on here, and these are going to check that the system's working correctly. We're going to give people medication to lower blood pressure, and we're going to give people medication to raise their blood pressure, not at the same time, but separately. Now, you might think, why do we need to know this? Why do we need to know that this medicine works? It's not that we need to know the medicine works. It's we need to know that the measuring devices are working correctly. If we give a person, or we give these 6 individuals, blood pressure lowering medication, and then we test them and their blood pressure did not change, we can probably hypothesize that maybe our device, maybe the person taking the blood pressure is doing it wrong. Maybe their machine's not working correctly. We know that something within our system is not working correctly. Same as when you give the individual's blood raising or blood pressure raising medication. If it doesn't raise their blood pressure, it's not that it might, it probably means that their blood pressure is being raised, but maybe the system that we're using to take the blood pressure isn't working correctly. So these are positive control groups. They're checking to make sure our devices and our measuring is actually accurate. Now, again, this is something you've probably seen back in middle school and maybe even elementary school, but I'm sure you've seen it in culture, biology, physics, chemistry, whatever science classes you took before AP Bio. But you have your independent versus your dependent variables. And we're going to talk a lot about these this year, but you should probably know about them already. The part of the experiment that you change, this is one variable. This is the one piece of the experiment that you change. That's called the independent variable. Again, we only test one variable. If we have more than one variable, we can't definitively say, well, it's this variable versus the other variable. Like the coffee experiment, we can't if we just did coffee versus somebody not drinking coffee. There are several variables. They're intaking something that's probably warm, that has liquid, that has water, that has caffeine and other things. We have to only test one variable at a time, and it's really important that we only test one variable at a time. Dependent variables. This is what is dependent on the independent variable. Wow, Mr. Friedhoff, I know this is kind of groundbreaking stuff, but truly, dependent variables rely on and they change as a result of your manipulation of the experiment. Now, the dependent variable usually goes on going up and down the y-axis. The x-axis is the independent variable because, again, that's the one we change, and we're going to chart how that changes over time usually. Next on our list is theory versus law, and you might be confused on when to use these two terms. Let me explain. Theories describe how and why things happen. Laws describe what happens. So laws only describe what is actually happening. It doesn't actually explain how and why those things are happening. That's a theory's job. Again, theories describe how and why things happen. Laws describe what happens. Theories are able to usually model and you're able to predict things. Laws are more of like an explanation under certain circumstances, and they're usually mathematical based too. Like F equals ma, that's a law. Now, down at the bottom there, you can see the theory of gravity. That describes how does mass have gravity. The law of gravity describes masses have an attractive force. They're saying what is happening. Theories describe how and why things happen. Now, you may have heard of somebody say, I'll accept the theory of evolution when it becomes a law of evolution. Now, that is a misconception. They think that laws have more evidence versus theories, and that's not the case. Theories do not become laws with more evidence. They will never become a law. A theory will never become a law because they explain two different things. Theories describe how and why things happen. Laws describe what happens. Now, here's a cool way on how to remember theories versus laws. Theories describe how and why things happen. Laws describe what happens. I love it. That was just by coincidence, but theories describe how and why things happen. Laws describe what happens. Now, moving on, belief. This is a claim not supported by evidence. And in this class, I always, guarantee that I will not force my beliefs on my students. One of my favorite beliefs is aliens exist. I think there are aliens out there, but there's no scientific evidence out there that supports the existence of aliens. Now, you could argue like, oh, there's been so many, abductions. There's pictures, there's videos. Those aren't falsifiable. Those aren't in a scientific like an experiment. We can't use those as data. They have to be in a controlled experiment. So we don't have any scientific evidence that supports there's aliens, but that's a belief that I have, and I would never force that on my students. Religion is a belief, and no matter what religion you are, no matter what religion I am, I'm never going to force my religion on you, and I'd hope that you wouldn't force your religion on me as well. This is a science classroom. We separate the two. We separate beliefs, hypotheses, facts, theories, laws, and we understand the difference between them. But just by the separation of church and state, we don't go over religion in my class just because it's not the proper setting. I love using this cartoon in class. I'm going to explain it here and read it to you. It says, there's an onion in space orbiting the sun. It says, prove me wrong. So you can't prove a negative. It says, you can't? No, find the onion first, then work backwards. And he says, well, now you've gone and done made proving stuff hard. Yeah, almost like it's a science. This is a perfect example of how science works and how people think science works. This person is saying, well, you can't prove something that there's not an onion orbiting space, which means it's true. No, you can't start out saying that. You have to support your hypothesis first with evidence. He says there's an onion in space orbiting the sun. The burden of proof is not on the female there to prove to disprove. He is making a claim. He has to bring evidence and support that claim, because no amount of evidence on her side can really ever disprove that. Maybe to his liking. She can say there's satellites out there and they're taking tons of video and pictures, but he say, well, it's just not enough. You just can't see it with your instruments. He is making a claim. He has to support it with evidence. This is how science works, and it's really tough. Now, the last section here we're going to just quickly go over is the scientific measurement. We've talked about this before. It's a base 10 unit. For some reason, it's like 3 countries, and the United States has to be one of them that doesn't use the metric system. But the metric system is involved within the SI system. You can see there it says volume is measured by liters, mass is measured by grams, length is measured by meters, and temperature is usually measured by either Kelvin or Celsius. But this base 10 unit is so easy to use, so easy to memorize, and I'm sure you've used it before, so I'm not going to really talk about it. I'll probably show a couple memes. Yeah, the United States, for some reason, just doesn't like the metric system. Hopefully someday, cross your fingers, we'll move over to the metric system. The metric system has 1,000 meters is a kilometer, and they have 5,280 feet in a mile. There's so many random numbers out there. I wish we would just go to the metric system. I think it shows you here. Imagine if Americans switched from pounds to kilograms overnight. There would be mass confusion. Good one. And in the United States, you can see inches to a foot, feet to a yard, yards to a mile, ounces to a pound. Like, they're just all over the place. Again, the rest of the world goes by 10. It's just so easy. And then even with the month, day, and year, I mean, if you ever look at international dates, they'll go day, first, month, and then year. It makes so much more sense. All right, that's the end of chapter 1. If you have any more questions, just shoot me an email and I'll try to get back to you, but I hope this helped.